change. But also at 10x Genomics. So we have launched a lot of new solutions, both at the single cell as well as for spatial applications, as well as the new instrumentation. So I have the pleasure to have Nirav Patel, our Science and Technology Advisor today, to highlight some of the most recent research that has been done using 10x technology for single cell and spatial, and also really focusing on multiomics. If you start looking into single cell and spatial papers right now, you'll notice the trend, which is more going into multimodality analysis from the same cell or the same tissue, if you're looking at spatial. So Nirav is going to highlight those and also tell you a little bit about the new applications and what's coming up over the next few months. So again, thank you very much for coming. Thank you for being on the Zoom. And at the end, we'll have a Q&A session. If you have any questions, feel free to raise your hand or type the question on the chat. And we'll also be here over the next day and a half with Nirav on campus. So if you want us to stop by and chat with you about your project or your interest, feel free to let us know. All right, Nirav.
Thank you, Fred. Okay, let me go ahead and get this out of the way. All right, perfect. So thanks and welcome everyone. So my name again is Nirav Patel and I'm a Science and Technology Advisor. And I think some of you might have seen me doing these talks before the pandemic sort of hit. I did several lunch and learn sessions. So I'm very excited to be here after this long 19 months, hiatus where I've just been behind this virtual, podium. So today what I'm going to be talking about is multiomics for the most part and how the single cell and spatial technologies are being used to generate novel insights into biology. So in my talk today, this is a brief outline where I'll start with the history of biological analysis. We'll also look at introduction to next generation multiomics using our Chromium and Visium platform. And I have some interesting stories that we'll cover where multiomics has been used in the context of COVID-19, oncology, and cardiovascular disease. And in the second segment of my talk, I'm going to be introducing Chromium X, which is the next generation of instrumentation from 10x Genomics, and then also introduce to Visium Spatial Profiling. So I'll start with the history of biological analysis. So if we look at the history of biological analysis over the past 60 years, what we realize is that the better tools that we have, we can address some of the questions that we weren't able to with previous set of tools. So for example, if you look at cell sorting back in 1960s, this was the first introduction into single cell analysis where we could take a set of parameters and start understanding cellular properties. And over a period of time, with incremental improvements in imaging techniques, laser technology, biochemistry, and now microfluidics, we've been able to develop some trends, that is higher resolution, more parameters. For example, in 1970s, FACS-based sorting became available. And this was a very powerful tool for immunologists to understand individuality of those immune cells, but with a limitation that you were just able to do 12 to probably now 30 marker or 30 parameters. And then for the next 30 years, there was some incremental improvement till mass cytometry sort of came in, where now you could use antibodies that are tethered to heavy metals. So you could expand that multiplexing ability now to possibly 100 different parameters. Along the same time, next generation sequencing and the Human Genome Project sort of came into existence. And with Human Genome Project and NGS, we started realizing RNA to be a of genes within a given sample, which was bulk-based RNA-seq methods and the genome sequencing method. And then if we move fast forward and if we sort of look at recent times, single cell sequencing really generated this resolution revolution. And with the advance of microfluidic-based technology and 10x Genomics Chromium system, we can now look at single cells, define the heterogeneity in a very scalable way. And one other trend that Fred mentioned is emerging is multi-omic cytometry. The real question is that, can we identify thousands of analytes across the central dogma of life? And I think eventually that might happen at the single cell resolution. We're not quite there yet, but we can also already see indications where you're now able to get multiple modes of information from the same cell. And that's going to be the centerpiece of my talk today and center theme throughout. And when I talk about single cell multi-omic cytometry, it can be in many different flavors. So this is one example where we're measuring RNA using unbiased gene expression techniques with the technology that exists at ICBR. We could tag on barcoded proteins, and we can start to measure tens to hundreds of proteins from the same sample. But we're not just limited to here. We can go above and beyond now to also measure other modalities. And this is largely through our technology. So we have two technologies that are powerful and complementary. So we have our single cell Chromium system that allows you to characterize cellular heterogeneity through analysis of transcriptomic states, epigenomic states, and multiomic states. We also have our Visium technology that was launched back in 2019. So now, without having to dissociate a tissue, you can take sections, you can generate spatially resolved data sets. And both of these technologies have gained numerous accolades. But if you look at multiomics, really the ball got rolling back in 2019 when it appeared as the Nature method of the year. And last year, spatially resolved transcriptomic was named as the method of the year, where Visium data was on the cover of Nature methods. So with that, I'd like to give you a little bit of glimpse, just to catch up on some of the things and advancements that have happened at 10x Genomics. So we started out with single cell RNA-seq back in 2016, where this product really became a flagship product very quickly. And it was able to generate so many beautiful, papers, but some groundbreaking, discoveries over the next few years. And as you can see from this particular recent paper, with single cell gene expression, you can characterize cellular heterogeneity. So here you see two compartments. You see the blood and you see the CSF. You can start to chart the phenotypes across these two compartments, which is the most common denominator. Beyond that, you can ask deeper questions. For example, what are the proportions of each cell type within each of these compartments? And we can see that if you look at granulocytes, they're more prevalent in blood as opposed to CSF. So that's the most common question that you can ask. Beyond that, you can start to understand the functional states of these cells. You can perform lineage analysis to see who comes from who. You can also reconstruct developmental trajectories. Next year, in 2017-2018 timeframe, we launched the immune profiling assay. And this really allowed the immunologists to ask deeper questions about the T and B cells. So the immune profiling assay would still give you the transcriptomic information to characterize tissue heterogeneity, but you have an add-on option there. So if you are interested in the role of T and B cells, whether it's cancer, it's vaccine research or neuroinflammation, what is the clonotype diversity of those T and B cells? And you can start to get paired immune receptor information from that same cell. Now, as we looked, when I showed you the history of biological analysis, the immunologists have traditionally defined cell type and cell states by looking at surface protein profiles. So in 2018, we introduced feature barcoding, where you can use these antibodies that are tethered to oligonucleotide. So we're expanding the potential space or number of antibodies you can use within a given experiment. And now, we see routinely papers there where 50 to 100 antibodies are used in a given experiment. So you get the gene expression information. You also get the protein expression information. But as the name sort of suggests, feature barcoding is just not limited to looking at cell surface protein. You can also look at, say, capture single guide RNA by barcoding that single guide RNA for functional genomics applications. Or you could also tag a multimer to study antigen specificity. So just to recap, the immune profiling assay is really the next generation multi-omic, where we're generating potentially 4 data streams from the same cell. So you can characterize the heterogeneity of your tissue by either looking at gene expression, or you can do that by using the protein expression profiles. And when you do that, instantly what you can see is that if you look at this T cell population, which sort of exists as a blob, it's much more resolved when you add an additional modality. So you have like multiple perspectives to interpret, biology. And then you can also get information for immune receptors. So start looking at those clonotype, distributions by getting full length information for alpha and beta chain of T cells, heavy and light chain for the antibodies. And another missing puzzle for immunologists really is to then also understand, okay, I understand the VDJ, but what are the antigens that those VDJ bind to? So you could tag multimers. So in this experiment, you can see we have 4 different peptides or antigens for flu, CMV, EBV, and we can start to define some of the antigen specificity information as well. Moving into the pandemic time, 2019 to 21, we introduced the first true multi-omic solution, so which is our single cell multi-omic assay. So with our multi-omic assay, you can get 2 readouts from the same cell. So we can start to understand chromatin accessibility or the epigenome of the cell and the regulatory logic. And from the same cell, we can also get transcriptome information. So this allows you to build better regulatory networks and start to understand the programming of the cell. And in end of 2019, as we were just seeing cases sort of rise for COVID-19, we launched our spatial product, our Visium product for fresh frozen samples. And just a few months ago, we also extended this to now FFPE samples. So now enabling processing of those samples that are in the biobank for retrospective analysis. And I'm going to talk about Visium a lot more in the last part of my presentation. Just to give you a quick overview of the workflow. So whether you're starting with our Chromium platform or our Visium platform, this is a very high level overview where it follows 5 different steps. So for Chromium, you can start with a high quality suspension of either single cells or nuclei. We perform all the partitioning and molecular barcoding using these microfluidic chips and reagents. And the Chromium system is at ICBR. We then generate Illumina compatible libraries, and on the back end, we have license-free pipelines called Cell Ranger that does most of the heavy lifting of the data. And then Loop is a desktop application that can allow you to quickly gain biological insights from your samples. Now, if you're starting with Visium, we start with tissue sections. And these tissue sections are placed on specialized glass slides. We provide all the reagents. You're generating spatially barcoded libraries for Illumina sequencing. And again, on the back end, we have the Space Ranger pipeline and the Loop browser for data analysis and visualization. So with that, let's talk about science. Let's look at a few recent publications using multi-omic cytometry. And I think this first paper might be familiar to many of you. This is the CiteSeq paper that came out of the New York Genome Center, which was the first paper that showed demonstration of using those oligo tagged antibodies with the 10x system. And what they showed in this paper is that the CiteSeq data is highly concordant with flow cytometry data. So what we're doing here largely is changing the detection system from fluorescence-based detection to now sequencing-based detection, which is highly quantitative, it's robust, and it's accurate. So when you look at the proportions of cells between flow cytometry and CiteSeq, we see very high concordance. But also when you look at dynamic expression of a given surface protein like CD8A, again, you can see that we see very comparable data between flow cytometry and CiteSeq. And then these data sets are also very, very high parameter multi-omic cytometry data sets. So here I'm showing you some mononuclear cells. These are immune cells, and the annotation is based on gene expression as well as protein expression. But the flow cytometrists, they're used to looking at these biaxial plots. So we can project this data on a biaxial axis as well. So if you look at CD4 and CD8 expression here, when we look at this data, we don't need complicated gating strategy to figure out that this is the monocytic population. Also, when we look at this blue expressing cells. These are NK cells with dynamic expression of CD8A. Again, you can quickly say that these are NK cells, and they're very nicely resolved from the CD8 positive T cells, which is very important to resolve these population first and then resolve their biology. And we're going to come back to this in a little bit. But there are several advantages to using this kind of approach. So, and I'm going to lay out 3 of those. So the first one is that when you're measuring gene expression and protein expression from the same sample, you can start to understand the transcriptional dynamics. So in this first case, when we look at CD14 and CD16 monocytes, we see very good correlation between the gene and the protein expression, but that's not always the case. Whereas when you look at T and B cells and we look at CD4 expression and CD19 for B cells, what we see is that you see sparse expression of these analytes at the gene level, but very robust expression at the cell surface protein level, because expression can be bursty at times. And this allows us to have orthogonal measurements from those same cells. And if you look at the last example, sometimes cells can be transcriptionally similar. For example, the naive and memory population. And just looking at the gene expression information, it might be difficult to resolve closely similar cell states. So here again, incorporating CD45 specific antibodies, we were able to confidently then resolve these immune states. And just to show you a study, so this study comes from Bali Pulandran's lab and Prabhu Arunachalam at Stanford, and this is a COVID-19 study where they've taken a systems immunology approach, where they have PBMCs. Those PBMCs are from 12 age-matched subjects, or cases and controls, and the cases here have very wide disease severity. They also incorporated about a panel of 36 different antibodies in this particular study, and they were able to resolve the cell types and cell states. But moreover, they were able to identify the different proportions of cell types and cell states and find correlates of cellular and transcriptional factors that define the severity of the disease. And when they look at the myeloid cell types, they were much more prevalent in patients that had COVID-19. They saw enrichment of antiviral modules, and they also saw significant downregulation in the antigen presentation pathways. Now, if you also look at this particular plot here, I wanted to stress on this. Once they had done their differential gene expression analysis, they were able to come up with a panel of gene and proteins where they were able to look at the ISG specific signature. And when they compare that with the interferon alpha concentration with ELISA, what they see is that they see very strong correlation. And what I'm trying to tell here is not to replace ELISA with this kind of analysis, but it strongly recapitulates some of the signals that you see with standard immunological assays. But moreover, these studies are really powerful, and they can tell you what cells to look at and what genes to look at just because of this high parameter nature and rich data sets that you get. The second study is in the field of cancer. So this comes from INS Ephiantis, where they're studying B-cell acute lymphoblastic leukemia. And I'm not going to go into the biology of this study, but maybe talk a little bit about their approach. So they're looking at these malignant B cells. And sometimes these cancer cell types, they can be highly plastic. They can express multiple lineage marker. And again, this sort of approach can allow us to better define those cell types. So in the first pass of the study, they flow sorted for CD19 positive cells. They also buffered with some negative CD19 negative population. And when they started doing their analysis, they had a hard time resolving the NK cell population from the effector T cell population. So within their study, they just said that it's the TNK group. It's like in a game of football, you have the quarterback and then you have the wide receiver, and you just sort of lump them as runners because you don't have better metrics to sort of define that. These cell types are very specialized and they have specialized killer function. And I think there should be approaches where you're able to better define those cell types and then resolve their biology. So then they went in and they designed a CytSeq panel with about 60 different antibodies, and they allowed this approach sort of allowed them to better resolve cell types and cell states. But you can also see that across these patients, there is so much heterogeneity across these, analytes. So this again gives you additional data to resolve, more biology. Now, moving on, I'm going to add another layer of data here. So we talked about epigenome. So we have an ATAC kit that can also do epigenome. So here I'm going to show you a study, and this is in the field of cardiovascular research, where they've taken a really complex, multi-omic approach, where what they've done is they're studying, cardiac remodeling after myocardial infarction. And so if you look at their study design, they embed the tissue in OCT, which is one of the requirements for our Visium assay. They take a few sections for spatial profiling. They take a few sections to then isolate nuclei, followed by fan sorting of nuclei. And then those nuclei are split across two different workflows. So they go through gene expression and chromatin accessibility. And this study comes from Christoph Koop and group. And it's a fascinating study. So when we look at the data, our Visium generates H&E images. And from the same tissue, we're also generating spatially resolved data sets. So when you look at the H&E image, it looks rather homogeneous. So they asked, like, what is the underlying tissue heterogeneity? And since they had the single nuclei RNA-seq data set, they're able to integrate these data sets with Visium. So now you can see all these different cell types that are present in this particular tissue. Now, in the context of myocardial infarction, there is this area called the border zone. It's like the area where you have the most tissue, damage, where you see a lot of, necrosis, probably a lot of dead, cardiomyocytes. And then you have areas surrounding this where you might see repair, you might see inflammation, and you also might, potentially see quite a lot of heterogeneity in terms of cell types and cell states. But moreover, when we're talking about Visium, the question also comes, can we identify rare cell types within my tissue? So if you look at this cluster number 9, these are vascular smooth muscle cell. So in their single cell data set, it's like less than 1% of that population. But because they have the morphology data, they could ask the pathologist to identify regions where the blood vessels are. And what they find is that largely this particular signature of vascular smooth muscle cells surround those blood vessels. So which means that it aligns with the morphology that the pathologist sort of sees. But then the next question was, they're interested in cardiomyocytes. So what does the cardiomyocyte heterogeneity sort of looks like around the area of injury? And what they find is that there are two prevalent cardiomyocyte populations within this tissue. There's cardiomyocyte 1 and cardiomyocyte 2. And when we look at the GATA4, which is a lineage defining transcription factor, it's sort of expressed across the tissue. But if you look at another transcription factor, the NFE2L1, it largely is just highly expressed in this region of the tissue. And NFE2L1 has a role in protection against ischemia and stress. So you can already see that within a small piece of tissue, you have a lot of damage that has sort of happened. Maybe areas where you see a bit of normal tissue, and right around where the injury has sort of happened on the border, there's repair that's sort of going on. So within the same tissue, you can ask multiple different questions. But since they already had the single nuclei ATAC-seq data, they can also start to understand a little bit of that gene regulation. So when they look at GATA4 across the two cardiomyocyte population, they look at transcription factor binding, and they see similar transcription factor binding in both the cardiomyocyte population. But you can clearly see that in one of the cardiomyocyte population, the transcription factor enrichment is significantly higher than the other cardiomyocyte population. So this really gives you or adds that spatial context to understand cardiac remodeling in areas where you have injury, inflammation, as well as repair. And then in the previous study, they had to split the samples across two different workflows. So they split the sample through gene expression ATAC workflow. And then they had to use computational methods to then infer the linkage between those cell types. And this was largely the approach that was being taken before the multi-omic assay sort of came in. But this approach does have some limitations, and there can be some false positive, false negative, sometimes misassignment, because largely you're inferring, and the inference is as good as the assumptions that sort of goes into the algorithm. And to sort of resolve that, we had this multi-omic assay that was launched a couple of years ago now. And with multi-omic assay, we can get two readouts from the same nuclei. So here in this example, we have PBMCs. The PBMCs would go through nuclei isolation, followed by bulk transposition, and then the chromium partitioning and barcoding. And we generate two data sets. So here you can see on the top is the UMAP projection for the gene expression. At the bottom is the ATAC projection from those same cells. And on the right, then we're annotating these cell types based on their canonical marker gene expression profile. At the bottom, we're annotating these cells based on their transcription factor motif accessibility. And within the loop browser, we also have a functionality to transfer labels. So once you've annotated gene expression data, because obviously there's so much data for gene expression, it's much more robust in terms of annotation, we can push those annotation back to the ATAC space to then further resolve these different cell types that we have in the ATAC space. But by doing that, we can just generate better regulatory networks. And one of the example that I want to talk about is this application node that we have, where we're comparing tumor versus normal cells. So the first step in your analysis is you would be performing differential gene expression analysis to look at the genes that are enriched. The second is, because you have the ATAC data as well, you can start looking at the peaks to gene linkage, by which what I really mean is you have a cis regulatory element, and how does that regulate a certain gene? You can then start to look at the transcription factor motifs, and you can start building these regulatory networks, which are much more robust than inference-based models. And I wanted to just briefly touch upon this study because we talked about the central dogma of life and multi-omic, and this group here, the same group that also came out with the CiteSeq paper, recently used our multi-omic assay to get 3 modes of information from the same cell. So as you can see that they permeabilize the cells, they first fix the cells, they permeabilize the cells. They also stain the cells with a panel of antibodies. So in this particular case, they used over 200 antibodies for intra and extracellular proteins. So they're able to generate 3 modes of data using the multi-omic assay. Now, obviously, this is something that 10x is not supporting right now, but as you can see that our customers are already sometimes a step ahead from us, because for our multi-omic assay, we use nuclei as the input, but the feasibility has already been established by certain groups. So in the next part of my presentation, I'm going to talk about Chromium X, which is our new instrument, and the high throughput assays. And I'm also going to talk about the Visium as the last segment of my talk. So as we sort of collect data and curate data, and this is all done through this group in Europe, the Valentine Swenson group and Lior Pektar's group, where they're curating single cell data. And when we look at the trend, there's some things that we can pick up. The first is that more and more studies are analyzing more and more cells. And as you analyze more and more cells, you also see that you identify more cell types, more states, more clusters. But there's 2 issues with that. The first one is it's prohibitively expensive to scale up your experiment. The second issue is that these academic methods, they're hard to reproduce. So there's a need for assays that are more robust, but there's also need for more affordable single cell sequencing. And I think that's where the HT or our Chromium X sort of fits in, and I want to maybe build a case around that today. So Chromium X is the next generation of instrumentation. We just launched this a few weeks ago, and Chromium X provides you the highest degree of throughput flexibility, whether you have a few hundred cells that you want to capture, up to over 750,000 cells per chip. So it affords a lot of flexibility. It's also one-stop shop, which really means that it's backward compatible. So all the legacy assays can run on the Chromium X system. And especially when you have large projects, for example, these can be cell atlasing projects, these could be large CRISPR experiments, or these could be large immune profiling, experiments, where you really want to analyze a lot of cells, usually over 100,000 cells. You can start realizing economy of scale in a graded fashion. And I'm going to talk about that a little bit. But Chromium X also has several other improvements, like it's the most controlled microfluidics. It's also wired to the 10x Genomics cloud, so all the data is stored there. When something goes wrong, our troubleshooting teams can really get in easily, and it sort of streamlines the experience. But there's more than one thing about Chromium X that sort of stand out. The first is the flexibility. And as you can see, that Chromium X is capable of running all these different kit configuration that we have. If you're just starting out your single cell journey, you can start with our low throughput kit. And with our low throughput kit, per channel, you can capture from 200 cells to 1,000 cells. So it's a great way to get started. As you've optimized your sample prep, you optimize your pilot data, looked at that data, and as you're scaling up, the most go-to options are our standard kit, and they're all available on the Chromium X. And then finally, when you have specific projects that require very high throughput, hundreds of thousands of cells, you can realize those economy of scale sort of experiment where we now have several levers that we can pull to reduce the cost per cell that you're running through Chromium X. But the Chromium X is also compatible with everything that I talked previously, like these feature barcoded antibodies and multimers, CRISPR, so on and so forth. And when we compare the Chromium X datasets with our standard Chromium controller, because we're using the same biochemistry and next-gen technology, we see that across the 4 single cell assays that we have, we see very robust and comparable performance across both those systems. So what I'd like to do today is maybe talk about a study, a high throughput study on Chromium X, and in terms of biology, what we sort of found by increasing the number of cells within this experiment. So in this experiment, we have some bone marrow aspirate. So we purified some mononuclear cells. They were cryopreserved using our demonstrated protocol. And when we were ready to run the experiment, we stained the cells with universal antibody panel from BioLegend. So these are human universal panel, which comprises of about 130 antibodies that are already pre-titrated and comes as convenient pack. We flow sorted those cells for viability. So we have a 7AAD that we use to sort for live cells. And those cells were then run either on our high throughput chip N or our standard Chromium chip K. So for this chip, the maximum that you can capture per channel is 10,000 cells. Here it's 20,000 cells. So we shot for 10 and 20,000 cells across these 2 different chips. When we look at the performance in terms of median genes per cell and median UMI per cell, we see very comparable data across the sequencing depth across our 5' version 2 HT and the standard kit. Also, the correlations are very strong across those 2 different kits. When we start looking at clustering, then we aggregate the data sets, and since these are the same biochemistry and we can use cell ranger aggr function to aggregate these data sets, we don't see batch effects, or batch effects are really minimal. Because of the throughput, you can just see that the plots are a lot more denser for the high throughput assay. And in this particular experiment, since we already had those antibodies, we can then use the antibody and the gene expression profiles to further annotate these cell types and cell states. So we went from a blob that looked like very hard to resolve to now 7 different subpopulations of T cells. And these are all sequenced at about 20,000 read pair per cell. Now, where the throughput really comes into play is when you start looking at some of the stem cell and progenitor stem cell populations that can exist at very low frequency. So if we look at this tail on the left-hand side, we have all these hematopoietic stem cell and the entire lineage of several progenitors cell types. We see that the blue bars are much higher. So you have a lot more cells that are identified in the rare population group, so that you have better, robust statistical vigor because you have more cell types representing those populations. But since this is an immune profiling assay, we can also ask questions regarding the immune repertoire. So here, we look at the T cells, and these are all generated using a new visualization tool that we have called nClone, where each dot is a cell, and cells that are sort of which is more than 3 cells where really the clonal expansion is sort of happening. We see a lot of blues, as you can see here. So and blues are these TC memory and the effector populations. So we're identifying a lot more biology by gaining extra scale with these kinds of experiments. Another study where we wanted to look at a tumor sample. So we ran a 3' high throughput chip. And so on the left-hand side is the aggregated data. So here's the high throughput and the standard data. And I wanted to maybe pinpoint 2 populations. The first is tumor stem cell population. As you can see, we have a lot more cells representing that particular population. But if you also look at this population, which is ELF3 MCAM positive population, we have about maybe 10 cells here, as opposed to probably maybe 1 or none in the standard, again. So scale allows us to, identify some of these potentially, cellular biomarkers that we would miss with using, the standard kit for certain applications. And I do want to address, the cost part of it, because I think that's sort of essential. So as I mentioned earlier, that high throughput chips really come into play when you have a lot of cells, ideally more than 100,000 cells that you're trying to profile. So if we do the cost comparison, and if you're using the standard assay on a project where you need about 160,000 cells, including sequencing, that would be around $27,000. Whereas with a high throughput assay now, we're significantly reducing the overall cost of the project by about 40%. If you're using a targeted assay, you can realize even higher reduction in those cost price. And I do want to leave this thought that there's several levers now we can pull. And obviously, Fred and I are happy to sit down with you because it's always project specific. So if you have a project in mind, we can talk about that. And the last part of my talk, maybe the next 10 minutes or so, I do want to focus on spatial profiling and introduce the Visium platform. So maybe I want to start with a question. How many of you have heard about the Visium spatial platform that we have? All right, that's great. Half of the room. That's fantastic. And how many of you here use these techniques or sample types in your research? Okay, fabulous. Thank you. So with Visium, this is a very high-level overview. We're generating 2 orthogonal data sets from the same tissue section. So here you have a microscopy, readout of a human prostate cancer sample. This could be either H&E image or it could be immunofluorescence image. And then the same tissue would then go through a series of molecular biology steps to generate spatially barcoded libraries. And the second readout would look something like this, where you have tissue clusters and you have these spots that we're going to be talking about. But all the spots that have similar clusters, they have similar transcriptional, profiles. And so now you have a union of imaging, molecular profiling, and histology, and you can toggle across these different, modalities that you now have in your tissue. And since the launch of Visium back at the end of 2019, we already have several, papers. This slide is old. We now have close to 125. So we've seen a rapid adoption of this technology, which was available for fresh frozen tissue. And now it's also available for the FFPE tissue. So maybe I'd like to take a few minutes to show you this video. It's about 3 minutes, and it walks you through how the technology works. And then I'm going to touch upon some of the modules of the workflow for this technology. So at the heart of Visium technology are these specialized glass slides, and these have 4 capture areas. Each capture area is 6.5 millimeters square. So you can run 4 serial sections, or maybe 2 serial sections, or those could be completely 4 independent sections. Each area has 5,000 barcoded spots. So each of these circles that you sort of see here, these are 55 microns. Center to center distance is about 100 microns. And each spot is laid with millions of these oligos. And for those of you who have used our single cell technology, this should look very familiar. So the oligo has 2 barcodes. So there's a spatial barcode. You can sort of consider that to be like a zip code, where any molecule that gets captured there has the same sort of zip code. But then UMI is like a house number, right? So every molecule that gets captured also gets a unique barcoding information. And this is designed to capture poly A species. And this shows you the workflow between the fresh, frozen, and FFPE. And I'm not going to go into a lot of detail with this, but as you can see, that the Visium does not require an instrumentation from 10x Genomics. We have the glass slides, we have the reagents, and the instruments that are required, like microtome or cryostat, as well as the imaging system and the sequencer, they're largely present as part of pathology course or the genomics course. And across the workflow, there's also several stopping points that are included so you can work around your schedule. And this shows you the sequencing format. So Visium for FFPE is about 25,000 read pair per spot. For the fresh frozen, it's 50,000 read pair per spot. So per capture area for FFPE, you're looking at about 100 million reads, as opposed to a 250 million read per capture area for the fresh frozen workflow. And I do want to talk about a study in closing. This is a study that we did where we have a breast tumor sample. And the breast tumor sample was split across 3 different workflows. So we embedded a piece of tissue in FFPE fixed tissue. We also had a fresh tissue that also went with standard embedding, which is in OCT for standard Visium profiling. And then we had 1/4 of the tissue where we isolated nuclei so that we're able to integrate the nuclei data set with the spatial data sets. And when we compare the fixed protocol with our fresh frozen protocol, we see that the FFPE and the fresh frozen protocol in terms of number of genes that are detected are very comparable. In this case, this tissue is about 5-year-old fixed FFPE tissue. We're still able to get high quality data types depending on the underlying quality of the RNA, where we do have like QC measures to make sure that the RNA is of high quality. When we look at serial sections for fresh frozen and FFPE, again, you see very high correlation, whether it's within the sample or across these 2 different methods that we're using. The correlations are very high. When we also compare with the IHC data to look at these 3 biomarkers of breast cancer, we can see that the Visium data correlates very nicely with the expression of these particular genes. So in this case, we also asked the pathologist to annotate the regions of ductal carcinoma in situ, fat, fibrous tissue, and immune cells, because we wanted to look at the infiltration of immune cells. And largely, as Fred mentioned at the start of the presentation, that a majority of the Visium data sets or papers that are published are with single cell RNA-seq to enhance the resolution. And by doing that, we're achieving 2 objectives. So in this study that comes out of St. Jude from Paul Thomas's lab, this is a lung tissue that is showing an ARDS, or acute respiratory distress syndrome phenotype. They also generated single cell RNA-seq data, and they found this population of damage responsive fibroblasts. So what they wanted to see then is, where is the signature of this particular cell population on my tissue? And what they see is that largely that signature is sort of more prevalent on the left-hand side of the tissue than the right-hand side. The second way is also to have better cell deconvolution at each of the spots. And there are several algorithms like RTCD or cell to location, where you can build proportions of different cell type specific signatures that you have at every spot on the Visium array. And so here in this study, since we had the nuclei data set, we had a specific question about the B cell, and we wanted to outline a specific B cell differentiation process. So we integrated the nuclei data set with the Visium data set and asked the question that, as the memory B cells differentiates into plasma cells, where are signatures for those cell types? And what we largely see is that those signatures are outside of the areas that have been annotated as ductal carcinoma in situ for B cells. But on the contrary, we can also look at the T cells and macrophages. And for T cells, what we largely see is that those signatures are infiltrating the tumor. So you get, again, 2 orthogonal measurements, and which is very powerful for biomarker analysis, but also just to understand cancer at a deeper level. And then for both the fresh frozen and FFPE, we've optimized several different tissue across human, rat, mouse, and we maintain an inventory of all the different tissues that we have optimized. So if you're sort of interested, you can see if your tissue of interest is on that list. And largely, just to recap, the Visium spatial gene expression allows you analysis of whole transcriptome in FFPE samples or fresh frozen samples. The resolution is high, so per spot you get about 1 to 10 cells. It avails true discovery. It's an assay, so all the glass slides and reagents are part of the kit. In terms of the microscopy readout, you have 2 options, either H&E or immunofluorescence. We have data analysis pipeline. It's an efficient workflow. And largely, we do have exceptional support. And this really starts at the experimental design. As you're designing your experiment, Fred and I will be happy to sit down with you. And as you start with your sample prep, we can bring in the applications, scientists as well to assist you with some aspects of the workflow. And lastly, thank you so much for your attention. Like, this has been fantastic to be in person here today. And this is your local, field team. And I'll take any questions. Thank you. Yeah. Yeah, absolutely. So the minimum recommendation for the gene expression assay is 20,000 read pair per cell. But it's always, cell type specific. So as part of the output, when you look at, the HTML file, once the sequencing is done, we do generate a metric called sequencing saturation. So that tells you the underlying complexity that you're capturing. So usually, as long as you're around that 60 to 70%, sequencing saturation, I think that read depth should be enough. If you're lower than that, you always have the library to resequence it deeper. Yeah. So for multiome, the epigenome data is we're looking at chromatin accessibility. So it's ATAC data. So right now, we don't have any biochemical modification that we can sort of measure at this point. So we're really looking at open regions of chromatin and the transcriptome. Does that answer your question? That is true.
That is true, right?
That is true. Although, we're not specifically measuring methylation. We're just looking at the open conformation of chromatin.
Okay.
Yeah, absolutely. So with the multiome, we're isolating nuclei that would go through bulk transposition, and we would do the capturing, and we're generating two separate libraries, one for gene expression, one for ATAC. And on our gel beads, we know the pair of barcodes that barcode the gene expression library and the ATAC library. So bioinformatically, we're able to pair those data sets together. They are sequenced separately. That's right. All right. That's right. That's that's a fantastic question. So there will be some areas, between the spots that will not get, captured. But you still have like the ability to get data points on up to 5,000 spots. If you are using immunofluorescence as a readout for your imaging, you can see what you're sort of missing between the spots as well. So that might be another orthogonal way to just capture some data. But having said that, I do have to mention that we are coming out with Visium HD products next year. So Visium HD will significantly reduce the spot size. So it's going from 55 microns to 4 microns, and then center to center distance would be 5 microns. So it will eliminate majority of that white space. Fred? Yeah, so the question is whether we can do feature barcoding with our VCM assay. We are working towards that. So hopefully in the near future, we'll have a supported assay to do that for our FFPE assay. And then, later on, we'll also enable that for the fresh frozen. There have been one group, we recently hosted them for a webinar where they've shown a panel of about 50 different antibodies that they were able to work with the VCM fresh frozen workflow. So it's similar to that, maybe slight nuances, but yeah. Yep, absolutely. Yeah. So the question is, let me know if I captured it right, whether the chips that you have for the standard assays would fit on the Chromium X system. Okay. Okay. Yeah, and the follow-up question is whether the chips that we have for the HT would fit on the standard Chromium system. Yeah, so yeah, that's a fantastic question. So the first part of it is that all the standard configuration of chips that we have currently will run on the Chromium X system. The second question is the Chromium X chips, will they work on the standard Chromium system? The Chromium X chips have 16 channels as opposed to 8 channels on the standard Chromium system. So in terms of hardware, the Chromium X does have new set of hardware to enable more samples, twice the number of samples. So those chips would not be compatible with the standard Chromium system. Yeah. Okay.
There's a couple of questions in the chat.
Yeah, so let me. Yeah, so the question is, in what kind of situation should we use gene expression from nuclei? So another, fantastic question. So the first I would say is your sample type, how the sample was stored. If the sample is frozen and archived, then really we don't have a way to dissociate those samples into a suspension of single cells. So nuclei sort of becomes the go-to for those, kinds of sample. The second is cell types that you might be interested in. Certain cell types can be a lot larger, like adipocytes or hepatocytes, where our microfluidic channels are about 50 microns in size, and we've tested cells as large as 30 to 50 microns, and they just work fine. If your cells are larger than that, it tends to clog our channels. So that's where nuclei isolation might be a way to go. And the third is sometimes certain cell types, they don't respond very well to stress. A good example of that would be podocytes from kidney. So as you sort of dissociate them, you tend to lose those particular cell types. So in that case, maybe it might make sense to go to nuclei so that you can improve the representation of those cell types. So another question that I have is, you mentioned that you preserved human breast tumor cells for 5 years. Was this sample in the fixative, and at what temperature? So that's a great question. So for the human breast cancer data set that I showed, the fixation was FFPE based fixation. So we're really looking at the fixed samples at that point embedded in paraffin blocks, and those were PFA fixed. They were just stored at room temperature for the most part. For the single cell part of that project, those samples were actually fresh frozen, so they were just stored fresh frozen in the freezer for that period of time. Now, I do want to maybe add a little bit more detail on the FFPE part of the workflow. So I showed you data from sample that was about 5-year-old, but at 10x we have tested samples that were freshly fixed to samples that were as old as maybe 13-year-old, FFPE fixed sample. As long as the RNA quality where we measure BV200 value is more than 50%, those samples should still produce robust data. Right, so I don't have any more questions online here. Well, thank you so much again for attending today. And please, let us know. We're here, like Fred said, for the next day or so. We'd love to sit down with you. Thank you.